Hello, everyone. Welcome to the podcast. This is Pavan Mirla. Today, we have Professor Luke O'Neill as our guest. He is one of the world's leading immunologists. He holds the Chair of Biochemistry at Trinity College Dublin. He is a Fellow of the Royal Society and ranks in the top 1% of the most highly cited researchers globally. Beyond his groundbreaking discoveries in innate immunity and inflammation, which Professor will walk us through, which have led to major breakthroughs in drug discovery. He is also passionate about science communication. You might know him as a regular radio broadcaster, host of the Show Me the Science podcast, or as a best-selling author of his hit books like Never Mind the Bollocks, Here's the Science, Immunology, and Keep Calm and Trust the Science. Please welcome to the show, Professor Luke O'Neill. Professor, could you tell a little bit about your research? Now, our audience is a student, maybe a professional who is passionate about making a difference to the society, either through research or something else to do with immunology.
Yeah, happy to answer all that. Very happy to be here, Pavan. Thanks for the invitation, by the way. I'm always happy to talk immunology or science, you know. So, well, I'm an immunologist. Yeah, that's my profession, I guess. And that's all about the immune system, obviously enough. And the immune system is extremely important, as everybody knows, I hope. Defends us from diseases like viruses, bacteria, cancer. Even the immune system can target tumor cells, you know. And I work on some aspects there, but my main focus is when the immune system goes crazy and it attacks our own tissues. These are called autoimmune diseases, is the word we use for them. It's things like Crohn's disease in the gut, MS in the brain, even Parkinson's and Alzheimer's. They involve the inflammatory process. There's a whole range of these inflammatory conditions. They're very difficult to treat, Pavan. There are some treatments. We must do better. And I guess what we do is figure out what's going wrong in the body during those diseases. And we know it's the immune system that's going off kilter. And ultimately, of course, we'd like to design new therapies to block these things that go wrong and treat patients. Our ultimate goal is to get better treatments for a huge number of diseases that afflict people that are still very serious.
For someone like me who is very new to immunology, I try to put up a few screens.
Yeah, these are great, Pavan. Yeah, so I guess if I go back to maybe 40 years ago, Pavan, I apologise if it's a long time ago, but I did a PhD in London in the Royal College of Surgeons on things called cytokines. On the right-hand side of your diagram, you've got these pro-inflammatory cytokines. And these were not known when I began my PhD, right? And gradually, immunologists discover them, and they're very important signals. They wake up the immune system, they drive inflammation, this process, and these are the very things then that seem to go out of control. And the first big breakthrough was to block some of them. So, for example, there was one called TNF, the IL-1 you have in that diagram there. These are very important drivers of the inflammatory process, and they cause disease. And we see a big advance beginning in the early 90s of therapeutics that could block them. And they begin working in things like rheumatoid arthritis, for example. That's an inflammatory joint disease. And there's been big advances there, and certain drugs will help people with that type of disease. And next thing then, we work on what turns them on. And these TLRs that you have there at the top, TLRs, they are the on switches. They trigger a signal transduction cascade, as you have it there, and that turns on the genes that then code for the cytokines. And my lab discovered some of the component parts there. You've listed MAL there. That was 2001. We discovered MAL. That's a key on switch that turns on this cascade of events and gives rise to these cytokines being induced. And of course, initially they were discovered in response to infection. So, for example, bacteria will turn on these TLRs. So will viruses. They turn them on. In many ways, they're like the sensors of infectious agents. And inside the cell, then, they trigger production of these cytokines. It's a bit like they're all messengers in a way, you know, and the cytokines are the messengers. And then we discover they also get turned on during autoimmune disease as well. It's a big unknown question, actually, what turns them on without infection. But we know tissue injury gets sensed by these things and promotes more inflammation. And you get a kind of a positive forward loop that's driving this whole process forward. So one of my first discoveries was how these TLRs work. And then very importantly, Pavan, they're part of the innate immune system. Now what that means is they're built in. They're in your system when you're born. They're innate, you know. And that was a big discovery because at the time, that innate immunity wasn't that well understood. There were lots of mysteries there, you know. But suddenly the innate immune system became a focus for many labs. Wins the Nobel Prize in 2012. for Jules Hoffman and Bruce Beutler. They were pioneers in this innate immune world, if you will. And my lab helped to discover some of the ways in which these things work. So in some ways, it's a very simple system at one level. You get an infection, you damage tissue, the products of these things are sensed by TLRs. You turn on MAL, you turn on the genes, and you make loads of cytokines. And the cytokines run away. Nice diagram for that. They run away all over the body. They turn on inflammation in different parts of the body then and drive the inflammatory process. And this is the very system we think that goes out of control in these inflammatory diseases.
I see. And Luke, so this purple circle, as I understand, is the immune cell. So this is part of the blood, right? So it is in the blood.
Yeah, that's a macrophage.
Okay.
We mainly work on cells called macrophages because that means big eater. So the macrophage eats stuff. It eats bacteria, it eats damaged tissue, and when it eats it, it triggers this pathway, and then we get the whole inflammatory process being driven. It's in the blood. It's also in your tissues, though. There's macrophages in your lungs, in your brain even, in your gut. There's various types of macrophages. The ones that live in the tissues are called tissue macrophages. And the ones in the blood they're called monocytes in the blood, actually, and macrophages in the blood. So you do see them in different parts of the body.
I see. Okay. And Luke, could you take us back on how you invented TLR? No, what was your inspiration? What was the moment you felt you invented something?
Well, I didn't discover TLRs. I discovered MAL, right? So, but they were discovered, interesting, in fruit flies. Can you believe that? The first description of toll and TLR was in fruit flies, of all things, right? And how flies defend themselves. A guy called Jules Hoffman, he discovers them in fruit flies. And that was the first look at them, right? And then we all see them and we can begin to study them, And then Bruce Beutler, he discovers one of them called TLR number 4. That's in humans. And that senses bacteria. So other people found them. What we did was how they work. I guess my job was to be like a car mechanic, I go under the hood, as it were, in the macrophage. And lo and behold, we find MAL. And MAL is a key relay switch that gets the engine going. And the engine is driving these cytokines, and that then drives more and more inflammation.
I see. Oh, nice. Thanks for sharing that. All right. I put one more slide on NLRP3 inflammasome. So it was very hard for me to understand, and thanks to AI, that it put a summary visual. So could you please help us understand this? What is NLRP3 here?
I can indeed. So these TLRs on the last slide, these toll-like receptors, they're discovered first, right? And people like Hoffman and Beutler and so on, they're found first, okay? It opens up the window on lots of these receptors and sensors, and we all begin looking for more of them, And NLRP3 was discovered by a guy in Switzerland called Jörg Schopp and a guy in California called Hal Hoffman. They report on another sensor called NLRP3. And this one becomes really interesting, Pavan, because this is inside the macrophage. It's actually living inside. So if stuff penetrates the macrophage, NLRP3 now senses those things that are inside, right? And that will drive inflammation. It drives IL-1. You have IL-1 on that slide. That's a very important output from NLRP3. It also triggers the macrophage to explode. So on the right-hand side there, you've got the macrophage bursting, right? And when a macrophage bursts, it releases lots of noxious things that are very inflammatory. It's almost like as if you've exploded some building, and all the debris is falling everywhere. And that's now driving inflammation. So NLRP3 will drive that type of cell death. The cell is dying. But even more importantly, Pavan, it's sensing things like cholesterol crystals in the blood vessels. It's sensing a protein in the brain called beta amyloid, right? Now, beta amyloid drives Alzheimer's. And the basis for this is NLRP3 gets turned on by beta amyloid, the cell explodes and damages the brain, Another one is alpha-synuclein in Parkinson's, another factor, this time in Parkinson's disease. So all these diseases like Parkinson's, Alzheimer's, heart disease with cholesterol, you may know about cholesterol crystals in heart disease, they all go through NLRP3 and they cause inflammation that damages the heart, cause a heart attack or damages the brain, So NLRP3 suddenly became extremely interesting as a sensor of these things that are building up. Pavan, one way to think of it is, imagine heart disease for a minute. All the cholesterol is building up, right? The macrophage goes in, sucks up the cholesterol, drives NLRP3. There's too much cholesterol, the whole thing explodes and now damages the heart, Now what that meant, Pavan, was if we could get a drug to block NLRP3, NLRP3 inhibitor. We call these things inhibitors. If we could discover a drug to block it, that could be a really interesting therapeutic. And my lab in 2015 discovers the
Mm-hmm.
first really specific NLRP3 inhibitor, a drug to block it. And now our drug's in the clinic.
Mm-hmm.
It's been tested in heart disease. It's been tested in Parkinson's. So it's a strange sort of
Mm-hmm.
circuitous route, you might call it, from the discovery of NLRP3, its role in different diseases, and now
Mm-hmm.
drugs to block it that will relieve some of the symptoms of these diseases. So NLRP3 is a very important kind of overall sense, you might say, of damage. And then blocking it might stop some of the damage
Mm-hmm.
that's ongoing after the initial damage. You're stopping the further
Mm-hmm.
sort of spread of damage in different tissues, depending on the disease.
See, amazing. So they're all interlinked.
They
One
are interlinked. It's
cause
very
for
complicated.
so
We
many
need AI for
problems.
that, to be honest, because these are very complicated systems, really. The ultimate proof will be in patients. If we block them, will we see a
Mm-hmm.
therapeutic effect? And then we know this is relevant to human disease. And at the moment, there's clinical trials running on these different approaches.
Mm-hmm. Looks like AI would be a perfect fit and very timely. These are quite
Yeah,
complicated,
yep,
multidimensional, non-linear, and
yep.
things are changing all the time.
You've got it. You've got it. And that's
Wow, I
happening. We're
think
using
what a
AI
great time.
to unravel these things. First
Mm-hmm.
of all, identify the key parts that are important. We'd love to use AI to predict which drugs might work, of course. That'd be marvelous because that would speed up clinical trials and the testing of these things. So we need AI more and more really to help us.
Mm-hmm. Yes, Professor. Uh, and the third one is
Immunometabolism.
immunometabolism.
You've got it. Yeah.
Metabolism, and in your research citations spoke something about itoconate and
Yeah.
sacchinate, which I didn't
Well,
understand
you're
much.
talking about my
Please
favorite
help us.
things, Pavan. My favorite things. Yeah. And this is funny. So about 12 years ago now, I suppose, we noticed in the macrophage. We look at the purple cell. There's a macrophage. I said we noticed that when they get activated and get turned on, their metabolism changes. Now metabolism to a biochemist is how you digest nutrients. When you eat food and metabolism kicks in to digest the proteins in your diet, right? Metabolism also builds things. It builds new cells, and very importantly, you make a thing called ATP. Now, ATP is the energy currency of life, right? It's the battery that fuels everything, and that was
Mhm,
known for decades, by the way. And then we find, strangely,
mhm,
unexpectedly, metabolism changes in the inflammatory macrophage. You begin to get a buildup of succinate. That was the first one you have there, succinate. That builds up. We find that in 2013. That's actually very inflammatory. That's promoting these cytokines.
mhm,
But then, secondly, in 2018, we discovered itaconate is there to block the inflammation. It's a stop, as you have there, an off switch. So this is all very unexpected
mhm,
that metabolites will be having these extra functions outside their normal role.
mhm,
And lo and behold, metabolic change
mhm,
in macrophages is very important for the inflammatory state with succinate, but secondly, can block inflammation
mhm,
with itaconate. So suddenly a whole new world opened up to us of metabolites and metabolism explaining how these macrophages work, both in health and in disease. So this was kind of an unexpected discovery for many labs. And that was this field that emerged as a new field, really, Pavan, immunometabolism,
mhm,
metabolic change in the immune system, in
mhm.
this case, in these macrophages. So that was a really exciting development.
I see. I see. And how are they linked to NLRP3 here?
Yeah, well, IL-1, see IL-1 there. So IL-1 comes off NLRP3 as well. If you like, it's multiple switches being flipped, right? It's a bit like an atom bomb in a
Mhm,
way. You need more than one key to get the atom bomb to fire. So you
mhm,
need NLRP3 and you need
mhm,
succinate. And they both get flipped during activation. And now they drive IL-1 as one link.
mhm. I see.
Again,
So
complexity
your
reigns.
lab, mhm. So your lab innovated these
Yeah,
or
yeah, we
found them.
discovered a role for succinate and the connection to IL-1.
This.
And then we discovered a role for
Uh-huh.
itaconate suppressing all this. There were two of my many labs, by the way. We're not on our own. Remember, several labs, often at the same time, are making very similar discoveries.
Mhm,
And you get a build-up of information, and
mhm,
that's reproducible. We call this between labs. Many labs have now shown succinate to be inflammatory in different contexts. And when our paper came out, there was probably
mhm,
2 or 3 papers around the same time by different labs that
mhm,
discover these things.
mhm. I see. I'm curious, how do you come up with these names? Say Sakinate
Oh, it's
or in
deadly.
general pharma names?
Deadly. Yeah, it's impossible. It's like learning a new language, Favan. We apologize for this, by the way, to non-experts, because you need to learn. It's like you're learning French or Urdu or whatever your language happens to be, or Irish. It's impossible. The name, well, Stuxnet and Attack, they were discovered decades ago, and they were named by chemists. NLRP3 was named by your
Mhm.
chap. I named Mal. My lab named Mal. So whoever discovers it gives it a name. So it's funny. And you come up with names that kind
Yeah.
of mean something maybe. But often they don't have any meaning, strangely. They've
Uh-huh.
got some meaning, but you wouldn't tell what they do from their name. That's the difficult part.
I see.
Well,
Okay.
IL-1, Pavan.
Okay.
IL-1 means interleukin. Interleukin number one. That means between leukocytes. So macrophages are a type of white blood cell. As you may know, the white blood cells are called leukocytes.
I see.
And macrophages make IL-1. It talks to another type of leukocyte, hence interleukin. So sometimes the names have some rationale to them.
I see. So I was thinking maybe it's Luke, interluke. So,
No, sadly, I can't claim that. No, I wish I could claim it, but I can't. Uh, another set of molecules are called leukotrienes. That should be Luke O'Neill, really, but it's not. Leukotrienes for different reasons.
okay. I see. And finally, I couldn't get AI to generate a visual for this. Mitochondria
Yeah,
and your lab's discovery of controlling inflammation.
sure. Well, you're getting into deep water now, Van. Yes. But what that was is the mitochondria are making the succinate and the atacanate, okay? So they come from a thing called
Mhm,
mitochondria. Now, mitochondria, anybody who does biology would have heard of them. They were discovered as what's called the powerhouse of the cell, right? It's where you burn
mhm,
glucose
mhm,
and fats to make ATP. That was
mhm,
discovered in the 30s and the 50s. Great triumph of biochemistry. So when you eat glucose in your diet, or you eat fats or fatty acids, they get burned in the mitochondria, right? And the mitochondria makes
mhm,
this thing
mhm,
called ATP, and ATP is the batteries of life. But ATP, your muscles use
mhm,
ATP, your brain uses it as a source of energy. That was all known for decades. And lo and behold, the very same structure is making the succinate to drive inflammation and to make atacanate to suppress it. So there's a funny thing going on there. We're not quite sure why this is, but the mitochondria are very interesting in this regard. They're a remnant, Pavan, of
mhm,
bacteria, by the way, that invaded our cells 2 billion years ago. Can you believe it? So, 2 billion years ago,
mhm,
an aerobic bacteria, one that
mhm,
uses oxygen, climbed inside another one and they became
mhm,
mitochondria. Isn't it strange? So, we think there's something going on there. It's a bit like the
mhm,
bacteria in our cells are now driving inflammation in a disease context now. So, we're very interested in that concept. It's called
mhm,
endosymbiosis. And I could talk about that all day, of course, but that's one idea that we have. It's actually the bacteria
mhm,
that live inside your cells that drive the inflammation and disease. That's one idea.
mhm. Very fascinating. And I get what I see now is next generation will have AI tools to
Yeah.
investigate this further. So,
Yep.
so the apprehension of AI taking jobs, I think, is not right because there will be new
No.
jobs,
To me, Pavan,
much
it's
more
like
complicated problems.
well, it's a bit like we invented a microscope. It's a new microscope. So you can see life using AI very clearly now. Hopefully, that AI will be useful as to see life in a different way. And when the microscope was invented, what in the 18th century, they could see bacteria for the first time. With AI, we will see complexity and we'll try to unravel this complexity using AI. So I think we'll always need humans to deploy it and help us.
Hmm. Hmm. Exactly. So we need the driver.
Yeah, we do. You need data. I mean, these
AI is just
experiments
the
that we
tool.
do, they generate big data for about any experiment in my lab, a lot of data.
Mhm,
So we'll use AI to
mhm,
analyze and look for patterns and, certain trends might be there and statistical relevance. AI will be used for all that, I bet you. Or it is being used now for that stuff.
mhm, mhm. And I've not seen communication across domains. For example, pharmacology and finance, quant finance. I think that would be a wonderful thing where we could communicate. What tools we use. Maybe there is some
Yeah,
overlap in it.
definitely.
What's your thought on it? Have you seen any collaboration like that?
Well, you do see it with the drug companies. They use finance a lot, as you know. And my own companies, for instance, you got to raise the money, venture capital and all that. So we do see that. But scientifically speaking, why not? I mean, often advances happen because two fields come together that you wouldn't normally see, you know? And in fact, what happened in my case was the biochemists were the ones doing metabolism. The immunologists were doing the immune system, and they came together for immunometabolism, and suddenly these new insights emerge. So we often see fields that seem distinct begin to collaborate. And new ideas emerge on the margins then, you know, because people think of it differently and have different approaches. So we're very keen on mixing up disciplines. That can be a very useful thing.
And so, next slide, I thought of your contribution uh, to communicate issues with COVID. So I wanted to maybe briefly, I wanted to ask you, what was your experience like? Because it was such a stressful situation, a lot of misinformation and uncertainty. So how did you handle it as a researcher and as a communicator?
Sure. Yeah. Well. I mean, the communication bit goes all the way back to 25 years ago, man. I teach students. Let's start with that, okay? And I teach first year biology. That's just like talking to a lay audience, like someone who's no science, you know. So I was always interested in teaching people biology anyway, and I quite enjoyed it, you know.
Yeah.
And then the media got wind of me in the
Mm-hmm.
radio. This is like 15 years ago I'm talking about, you know. And I'd go on the radio sometimes and talk about the immune system or some new discovery about inflammation or whatever it is, right? So I was doing it anyway. And then the pandemic started, and the radio show I was on like once a week at that time, right? This is 2020. And they said, come on every day. I said, what, every day? This is outrageous. But of course, the reason is the appetite was massive. Everybody was scared, as you may remember. The science was happening in real time, you know. And I was honored and privileged to be asked to do it because I got great reaction. People wanted the information. They're very hungry for it, you know. And because I'm an immunologist, it was
Mm-hmm.
my business, you know. So I knew all about viruses. I knew about the immune system. I knew about inflammation. COVID is an inflammatory disease of the lungs, you see. And I knew all the mediators and all the stuff. NLRP3 was found to be sensing the virus, for instance, you know. So it was easy for me to use my expertise to do it. And there was never a more important time, was there, Pavan, really? Because the world was in turmoil. In Ireland, people were very scared,
Mm-hmm.
especially over 60s. The older people were scared because they were high risk of having severe disease. And my
Mm-hmm.
mission suddenly became,
Mm-hmm.
instead of just informing people about interesting science, it was informing them of a really important thing in their lives, you know. So it's very rewarding as a result. And I must
Mm-hmm.
say I got a huge amount of satisfaction out of helping people. And then the vaccines come along. I could explain them. I could tell people
Mm-hmm.
the evidence behind them. I could talk about clinical trials and reassure people. Now, the
Mm-hmm.
misinformation had to be countered for that. There was a lot of misinformation out there.
Mm-hmm.
It was the biggest mystery to me was how could a mask be political, for God's sake, in America? It was outrageous, you know. But I was quite happy to give the other
Mm-hmm.
side and give the scientific side and give the evidence. It's very important to talk about the evidence for these. If you're a
Mm-hmm.
scientist, you're basing what you say on
Mm-hmm.
evidence. Remember, Pavan. During that time, someone gave
Mm-hmm.
me a T-shirt, right? It said, "Without data, you're just someone with an opinion." You know, so I could cite the data behind these things. And for the first time in a long time, science
Mm-hmm.
became so central to people's lives. So I was honoured and privileged to be able to give
Mm-hmm.
my expertise in a very important at a very important time.
Mm-hmm. I think that was a great fit of someone who knows the topic and trying to
Yep,
balance that emotion versus science.
exactly, exactly. And I've learned Pavan over the years, beginning with students, how to convey this in a way that's understandable.
Mm-hmm.
You know. And people aren't.
Mm-hmm.
I mean, the big thing I learned was most people are very smart. Remember, so there was a way to do it that wouldn't
Mm-hmm.
talk down to them or shy away from complexity. You could explain it in a way as long as you use analogies. You got to use analogies and various ways of explaining
Mm-hmm.
stuff. And then people because it was a bit like talking a foreign language. Like as we just said,
Mm-hmm.
NLRP3, like ACE2 is the receptor for SARS-CoV-2. Already you've lost them. You know. It's simply because the words are so difficult.
Mm-hmm.
So I had a way to sort of make
Mm-hmm.
it understandable. I guess was the secret.
Yeah. No, I completely agree with that. When we communicate complex topic and we get more questions, that means the audience has understood it.
Yeah, exactly. Yeah, the
when
trick, the
when I
trick,
don't get questions, I know that they didn't understand it.
they didn't get it. They didn't get it. Well, you can intimidate people as well. But the professor always talking about all this complex stuff that puts people off. I think the trick was as well, Van, be very careful with technical words, because if you use it early, they stop listening. We all do, don't we? The jargon puts people off. So in each interview on the radio, I'd use
Hmm.
one or two maybe and explain them, because otherwise people stop listening. There was a trick. I
Hmm.
learned a trick, how to not be too jargon filled. That really puts people off.
Hmm. Yes, Professor. Yeah. I use analogies everywhere, and that helps
Yeah,
me relate
you
more
got to
to
use
it
them.
and ask
You got to
questions
use
about
them
it.
because people can relate into their
Yeah,
own
it is.
lives. Then.
Yeah, exactly. All right. And Professor, we touched a little bit about science communication. So, who has been your favorite science communicator?
Oh, I can list loads of them. The great David Attenborough. It began with him, actually. To be honest, I mean, he was he is my, he's still alive, because one of my heroes. The way he explained the natural world was superb when I was a teenager. I'm sure you know David Attenborough, very famous. Another, Carl Sagan. Did you ever come
Mhm.
Sagan? There was a show called
Yeah.
Cosmos that had a
Hmm.
big effect on me as well. Those sorts of people really
Hmm.
inspired me. And when I was maybe 15, when I read a book by Isaac Asimov. Did you ever come across Asimov? He was a famous science fiction writer,
Yeah.
actually. But he wrote a book. He wrote three
Hmm.
books, one about biology, one about physics, and one about chemistry. I think it's Asimov's Guide to Science. And I read those books when I was 15, and they really blew my mind
Hmm.
because they described it so well and so clearly.
Hmm. I see. Oh, wonderful. Um, so, Luke, could you tell us a little bit about these awards, the Boyle Award? Because they mean a lot, no, but the audience, I don't know much about them. Could you tell a little bit
Yeah.
about those awards? Yeah.
I'm very happy to show off, Pavan. I'll show off now at the award. Well, it's always nice to win an award, I suppose, because you're getting recognised. But science isn't about awards. Never
Hmm.
forget that. Science is about just being curious and discovering things. The joy
Hmm.
of discovery is the real reward here. I'd also say that they're not just
Hmm.
for me, that's my lab, because I won those awards for our discoveries, right? And of course, the
Hmm.
whole lab is involved. Our PhD students, the postdocs, and I'm at the front of it. I'm like the manager of the football team in a way. I don't play on the field anymore. I wouldn't be in the lab much. I'm the manager. So it's very much a
Uh-huh.
team effort, and it's nice. I mean, the Boyle Medal is a bit in Ireland. That's one of the biggest medals you can win in Ireland for science. It's named after Robert Boyle. You may
Hmm.
have heard. He's a famous chemist. He was
Yeah.
Irish, Boyle.
Hmm.
He was actually Irish, you see.
Hmm.
So it was named after him. And then I got the Royal
Hmm.
Society. Was a big thrill to become a fellow, because all these famous
Hmm.
scientists are fellows of the Royal Society. Although as one of my friends said, "Man,
Hmm.
I got that FRS. That means you can get an FRS after your name." One of my friends said it
Hmm.
stands for former research scientist. You're past it. So that was quite nice as well.
Hmm. I see. I read somewhere that Ramanujan from India, no,
Yeah,
he is also FRS.
yeah. Oh God, yeah. There's some very famous Indian scientists, and the FRS was given to people in the Commonwealth as well. I think, and Ireland was concluded as part of that.
Yes,
He was a
yeah.
mathematician, was he?
I
Van?
see.
Was he a
Uh,
mathematician, or was he a biochemist?
yeah, yeah, a mathematician. Yes,
Mathematician,
yeah.
yeah, yeah.
No, he died too early. He died of tuberculosis, TB. He was just
Ah,
32 years
yeah,
old.
right. There's
Yeah,
another
there's
famous
an interesting
Ramachandran
story.
biochemist as well, by the way. There's a guy called Ramachandran who was a very famous biochemist. He was an FRS as well.
And the next one I'm curious about is your research to innovation. Are you still an entrepreneur?
Oh God, I am. Yeah, yeah. I'm very keen on that, Pavan, as well, because I mean, the very first disease I worked on was in 1985. It was on
Hmm.
Crohn's disease. You may have heard of Crohn's, it's the inflammatory bowel disease, right? And I kind of knew then, I chose it because there was a puzzle, like what's going wrong in that disease. It's inflammatory, it's in the gut, it's mainly in
Hmm.
your digestive system, gets attacked by your immune system. So I was doing it out of curiosity, but I knew we could make a discovery
Hmm.
that would help patients ultimately. This is a nasty disease. It involves awful diarrhea,
Hmm.
bleeding, all sorts. So I knew there may be something useful that might come out of it. The next one I worked on was rheumatoid arthritis. Began working on these cytokines. Big
Hmm.
drugs were discovered that
Hmm.
block these cytokines. And again, I would have been publishing a bit. I didn't discover the drugs, but I would have been publishing in the field. So I always had in the back of my mind, oh, maybe we can make something sort of useful for patients out of this. And when you're in a university, Pavan, you can get a
Hmm.
patent, right? So I had a couple of patents on
Hmm.
my discoveries, and then I began to form companies. And I was lucky enough to have three companies
Hmm.
actually. One of them is still going, Citrix. Now they're trying to target the immunometabolism. So what if we can stop the
Mm-hmm.
inflammatory metabolism? Wouldn't that be marvelous? I mean, our drug is in atopic dermatitis at the moment. It's a skin disease, to eczema. And then NLRP3,
Mm-hmm.
we found the first really specific inhibitor. And my company Inflasome was bought by Roche, the Swiss company, and they've just finished these 3 trials in heart disease, Parkinson's, and asthma. So I was always interested in that side of it to make the discoveries real, you might say. And that means forming spin-out
Mm-hmm.
companies, raising finance. We were backed by venture capital, and then doing deals with
Mm-hmm.
big drug companies. They've got very deep pockets. They can take things all the way to the clinic. So I always had that as a thing because my
Mm-hmm.
PhD actually in London was in pharmacology, immunopharmacology. That's the science of drugs of the immune that target the immune system. So I was always in that arena, trying to
Mm-hmm.
understand the
Mm-hmm.
therapeutic side of these things.
Mm-hmm. I see. Now, I wonder how someone navigates that wait period, because the innovation and the proof takes such a long time. There's a
Does
gap in between, a lot of failures. I wonder what's the thought process when something's fail in between, but you still persist. Could you walk us through what's the thought process of researchers and
yeah.
entrepreneurs in this domain?
That's a very insightful question, Pavan. Very insightful because it does take forever, right? First, okay, so we
Yeah.
discovered the NLRP3 blocker in 2015. We're in the clinic now testing it. It took
Hmm.
that's over 11 years. I guess you just got to accept
Hmm.
it. You got to be careful with the trials. Remember, look at safety. That can take a while. Picking patient groups.
Hmm.
As you may know, you go through 3 phases: phase 1, 2, and 3. So it just takes time. I think you can't depend on it. Let's put it that way.
Hmm.
You got to get satisfaction from tiny wins as you go along. And the tiny win might be,
Hmm.
for example, publishing a paper, say, or at a conference you give a talk. So what keeps you going is the tiny wins. Secondly, you got to be ready for
Hmm.
failure. Many of these things fail,
Hmm.
and you got to have stamina
Hmm.
and resilience is the word I would use. You often get that from your team,
Hmm.
from your collaborators. You keep each other going. There's a bit of camaraderie, as well. So,
Hmm.
so those things kind of keep you going. And my other big phrase is shots on goal. So at the moment, I would have 3 separate drugs that are different being tested. Maybe one will
Hmm.
work. You never know. One might work. It may not. I mean, that's why we need AI. The failure rate is 50% at phase 3. That's far too high. Surely AI could say, stop,
Hmm.
do this instead or whatever. I hope an AI will help speed up the process.
Hmm. Hmm. Awesome. Okay. And so, yeah, these are the questions that I had. Most of it we covered. Since we are short of time, Luke, so I wanted to ask you about the tools of the trade. What tools do you use for research, especially when you have biochemistry and such complicated
Yeah.
connections and relations and dependencies?
Yeah, you learn the tools of the trade, of course, when you're a student initially. You do practicals in labs, for instance. And then in your PhD project, you'll be
Hmm.
using, so we call them techniques, is the word we use for these things, right? The microscope is a very important instrument for us. We look down at it and see the macrophages, make sure they're healthy. And
Hmm.
then we use all kinds of techniques. We can look at DNA. The gene sequencing is a big technique for us. We can measure RNA. We can measure proteins. To think of mass spectrometry. You're using techniques of chemistry really often to analyze these things. Like to study IL-1, you can use high resolution chemistry to detect it and measure it. We call them assays, Pavan, interestingly. It's a bit like you're in the gold field looking for gold and you assay the gold. So there's a whole range of techniques that we use that people learn. We use cell culture. We can grow cells outside the body. It's called cell culture. We can grow macrophages in a dish and study them. We can use engineering to knock
Hmm.
out genes in cells, think of CRISPR. Often, Pavan, Nobel Prizes can be given for a new technique. Allows you to see something,
Hmm.
that you couldn't see before. So there's a whole range of methods that they're very elaborate, and I mean it's a bit like apprenticeship. When you do a PhD, you're learning
Hmm.
how to be a master. You know, like when you learn to be say a goldsmith, you learn how to work gold, don't you? In our case, you're learning these
Hmm.
techniques to deploy them to try and answer scientific questions. Then,
Hmm. Hmm. I see. Thanks for sharing that. And one quick question on the skills. So, because so much information is collected,
Yeah.
And in finance also, you come across something and you don't use it afterwards, it doesn't grow. You start again. Uh, so in terms of thinking and learning, so what
Yeah.
advice would you give to youngsters across domains?
Oh, yeah. Happy to answer that one. I'm telling my students all the time when how to do that. It does take a long time. I mean, to be a scientist, right? If you're a professional researcher, say, you want to go into a lab and be a real scientist, you get a degree, first of all, that can take 3 or 4 years, right? Now, in that, you're learning all about biochemistry or immunology or whatever your specialty is. You're learning about that, and it is complicated. It does take 2, 3 years to learn it. And then you're also learning some of these methods in labs as well. Then you do a PhD, or you can do
Mhm,
a master's, of course, and learn even
mhm,
more about something. And like in Trinity, we've got a
mhm,
master's in immunology, and some come in and do it. Their first degree might be biochemistry, but they want to do immunology, so they do an MSc in immunology. Then you do a PhD. Now, to get
mhm,
a PhD, Pavan, you got
mhm,
to be a real expert in the area, right? Number two, you got to do stuff
mhm,
in a lab, learn all the skills that we're mentioning, how to do statistics and everything, right? And number three, make a discovery.
mhm,
You got to make a brand new discovery. You're discovering something nobody's ever seen before in the world. That's the key moment for the scientist, is to discover something brand new.
mhm,
And it's such a thrill for that. You can imagine if you see something. When we found Mal, we were the first people in the world to see that. And then you publish your paper, other
mhm,
people then reproduce your work, and Mal became this big switch then in the macrophage. So you're kind of doing it for
mhm,
curiosity. That's the first trait you need. You need to be curious about things.
mhm,
And it can be anything. It can be dinosaurs. It can be stars. In my case, it's the immune system.
mhm,
So curiosity is the first thing you need.
mhm,
Then you need perseverance, diligence, get stuck in, go to college, learn all about this stuff, try and pass your exams.
mhm,
And then the big skill you
mhm,
need ultimately then is interest in what you're trying to do. And that can apply to anything.
mhm,
And if you're interested, you're more
mhm,
likely to succeed, aren't you?
mhm,
There's no point doing immunology if you're
mhm,
interested in finance, is there, for instance, or vice versa. So
mhm,
those are the kinds of things. I
mhm,
mean, intelligence is important, but to me, intelligence is often to do with application and trying to learn things.
mhm,
I really believe
mhm.
if anybody can do it.
Yeah.
Never imagine that you
Mhm,
can't. There could be someone listening into us now and they're thinking, oh, I'm not a scientist. All you need to do is get stuck in and learn. And for me personally, I had a great biology teacher in school. Fran Mooney's
mhm,
his name. He inspired me. So you need help from teachers and mentors to encourage you and help you through the process. But ultimately, you need curiosity, a bit of resilience, and a bit of stamina. And remember, Fran, the mission. In my case, the mission is to discover new medicines. That's the bottom line for these diseases. That's the ultimate mission here. Keep your sight on that mission. Don't put all your dependency on it because it's so difficult. But you're doing something very important. Ultimately, you might make a
mhm.
discovery that will ultimately help discover a new treatment for Alzheimer's or Parkinson's or Crohn's disease or whatever these diseases are. Your work will make a difference then.
Yeah, I completely agree with that, and I can relate to it. I started working on a calculus textbook,
Oh, did
and
you? Oh,
calculus
yeah.
was very difficult for very difficult topic for me. But I thought, with my average intelligence, if I can develop a book for dyslexic students, for autistic
Great idea.
students who have difficulty in understanding even to the level I understand, and as I started working on it, I found that wow, this can
Oh,
be
yeah.
learned if there is a mission to it.
That's right. You've got it. That's another good example, Fanny. And remember, for any school kids or young people listening in, I mean, often you're put off by the teacher not being great, actually, or maybe the mood you're in that day, or something arbitrarily stops you. Oh, I couldn't do maths, or I couldn't. You have the
Mhm,
ability if you just can unlock it, unlock it in yourself in some way. Then you can go and do it.
mhm,
And you're right that the mission that you had there was a really laudable mission with dyslexic. It's fantastic. I think ultimately that's what discovery might ultimately be about. It's doing stuff that helps our fellow humans. Really, ultimately, that's you get the biggest kick in the end out of that. And in my case, teaching students is a huge thrill. That in itself is good enough, really.
mhm. Yes. Awesome. All right. So we covered the research process also. One question on collaboration. So there
Yeah,
is this opinion that if I share my idea, it could be stolen. It may not be credited. So there is hesitancy in say finance to share
yeah,
insights that could benefit from collaboration.
yep. Oh, it's an essential question. I mean, it's human nature, isn't it, Pavan, anyway, to worry about that? We are humans, remember, us scientists. And you in finance, you're a human being, And that means there's rivalries and
Hmm.
trying to get ahead and get the promotion or whatever it is. You've got to remember that the
Hmm.
best way to make progress is through collaboration, because that other person might have an idea that you missed, for instance. In my case, they might have a lab
Hmm.
technique we haven't got, and we can use that technique. So again, you got to collaborate and
Hmm.
share the credit. There was a great phrase, Pavan, I think it was
Hmm.
Woodrow Wilson or someone said, "We'd make a lot more progress if people weren't looking for credit." It's funny. It's a funny way to think of it. That can stymie collaboration. But the good thing about the positive aspect, Van, is it's like being in a band, and we may come on to that in a minute. You're playing the guitar, this guy's playing the bass, and this guy's on drums, and this woman's a singer, whatever it is. That's a much better experience because you're all
Hmm.
collaborating, and the final sound is better then. So that's a good analogy of why we should
Hmm.
collaborate.
That's a great way to relate. The music is much better when there is a coordination.
Yeah, that's right.
Otherwise, it will be just one instrument.
Yeah, yeah, that's right.
Awesome,
Well,
awesome.
the solo artist
All
can be good as well,
right.
remember.
So,
But for me personally, being in a band is much better.
hmm, hmm, I see. Okay, so yeah, we covered these achievements, and congratulations, Professor. Glad that your achievements were recognized.
Thank you,
Uh,
thanks very much.
and and final question, Professor, on immunotherapy. In your
Yeah.
podcast, you mentioned to treat cancer, probably we should understand the immune system more. Could you help us understand? Because these days we see many cancer cases. Is there a hope where with immune therapy? Yes.
If you're an immunologist, it's very hard to avoid cancer because it's such a big disease. In my lab at the moment, we have one project on cancer. We've published a couple of papers over the years. It wouldn't be my main interest, is more inflammation. But it's central, really, because it was discovered as much as 80 years ago, Pavan. If you can get the immune system mobilized, it will kill the tumor, right? It's got great weapons. It's using these weapons to kill viruses and bacteria. The same weapons can be used to kill a cancer cell. And that was discovered, I suppose, 80. Coley actually in Boston was the first to say that. And what's happened now is extremely exciting.
Hmm.
It's the frontier in cancer is immunotherapy, getting therapies to get the immune system to kill the tumor. And they've been the biggest advances in cancer
Hmm.
recently in many ways, is mobilizing the immune system against tumors. So, for example, melanoma was a lethal disease. Now, 25% of people are cured with immunotherapy. And we're seeing, and we need to do better. It's still,
Hmm.
the overall response rate is 20-30%, but we can do a lot more there, and we can certainly boost that and get more and more people treated. And then the second thing is vaccines for cancer, and that's a really exciting area. And remember, a vaccine is you show the immune system a piece of something. It could be a piece of a virus, but you can show them a piece of a tumor. And now the immune system's trained to
Hmm.
recognize the tumor and goes in and kills the tumor. And that's a really exciting development, is
Hmm.
using cancer vaccines. So many of us feel that the immune system will be the answer in the end for cancer, because if you can just get it working right, you will kill the tumor in its tracks and stop it
Hmm.
developing.
Hmm. I hope this works because so
But
many
in fact, for
lives are
anybody,
cut
any younger
short of
people
this.
listening in, that part of immunology is a very exciting one to join, because we should get the response rate up, and cancer will no longer be such a fearful disease then.
Hmm. Hmm. Okay, Professor. Uh, that's a, so it's been such a honor for me, a pleasure talking to you, Professor. I really appreciate you making time. uh,
No
uh,
problem, Van.
so,
Very happy to
so
chat.
thank you, Professor. No, it was a pleasure
Will
talking
you
to
will
you.
you become an immunologist yourself? Come and do a PhD, Pavan, in my lab. How about that? If you're too busy with many other things.